Good morning, everyone. My name is Bill Weiser. I'm Executive Director and Global Head of Analytical Sciences for Patheon. And I'm here this morning at the AAPS conference in Denver, and I'm going to present a little bit about our integrated analytical development approach to pharmaceutical development. I'll talk a little bit about it, give a brief introduction to Patheon, talk about the rationale and value for integrated analytical development, and then also end up with a couple of examples where integrated teams did product development. To begin with, Patheon is really leading the transformation of the contract pharmaceutical development and manufacturing industry. We have a broad range of product solutions in terms of oral solids, sterile products, etc., which allow companies to focus on their core competencies. For instance, companies that do discovery or identify products or generic products, new molecular entities, etc., can focus on their core, while Patheon handles the flexibility to manufacture APIs, drug products, do the formulation development, as well as commercial manufacturing. We're basically building a better supply chain model by providing a full range of services. Patheon has been in existence since 1974, and basically throughout the years we've acquired capabilities that include soft gel technology, high potency manufacturing, solubility enhancement, complex formulations, and basically we cover about 75% of all dosage forms. We're also one partner for every step in the process. As compounds go through development, clearly they go through preclinical, phase 1, phase 2, phase 3, and on to commercial manufacturing. There's different requirements for that. Some compounds require a longer period of time to develop the appropriate dosage form in terms of solubility enhancement or something. And then in late clinical, really the focus is on transition to manufacturing, scale up, registration batches, and stability testing as well too. And finally, in life cycle management, in many cases, adding value to products by perhaps generating combination products, unique packaging or something like that can also be accomplished late in the development plan. But the purpose of this slide is to really demonstrate that Patheon covers this entire range all the way through from preclinical through commercial for large and small molecules, sterile and oral products. We also understand too that the current model of outsourcing really focuses on perhaps using a supplier you're not familiar with, and there are a number of handoffs, whether that's a handoff for analytical, for solubility enhancement, another area for scale up, and every time you have one of those handoffs, information is lost. So for instance, knowledge about the analytical characteristics of the compound, the formulation process, stability, every time you have that handoff, that you lose value. And so really by integrating all these approaches, we have the insight and the capability to really anticipate and resolve issues. So in other words, by engaging our product development teams throughout the process, we're sharing that knowledge, we're building on it rather than having to relearn on that in every step. And so what that does is that also facilitates the speed to market and also improves the financial performance as well too. As a matter of fact, 33% of the products that we have developed are also manufactured at Patheon. So again, a very strong pipeline of putting products through the program. Our Patheon is a global service provider, and so we have facilities throughout North America, Canada, Mexico, as well as in Europe and Australia. So again, a large global reach. Many of our customers have products which fit certain markets, and so the ability to actually have products spanning the globe is really quite important. On this slide here, I've highlighted our pharmaceutical, our drug product facilities, primarily our Canadian ones, Greenville and High Point, North Carolina, Oregon and Manatee, Puerto Rico, and then also our facilities in Europe that are focused on drug products. And I'm going to talk a little bit more about integrated analytical, primarily focused on our drug product capabilities. Traditional pharmaceutical product development and outsourcing is really been very tactical up and now. It's really based much more on a commodity mindset. I want to buy a white powder, or the drug product is a 100 milligram film coated tablet. I mean, that sounds good, but what happens is really in every case you're throwing the product over the fence. And so anything to do with the formulation, the manufacturing process, we're not connecting that information. It's disconnected. We're not benefiting from having that integrated. And so we're losing that value. Really, for pharmaceutical product development, it needs to be robust and scalable. And so this needs to take in the characteristics of the API, such as particle size, the salt form, polymorphs, et cetera, as well as on the drug product attributes and how the API impacts that in terms of bioavailability, delivery approach, stability, patient compliance, et cetera. And so really what we're after is making sure we have a strong feedback loop between drug product and drug substance. And being an analytical chemist, of course, the world revolves around us, and my point is that's usually mediated by analytical data. So having the information about the performance of the drug product and how that relates back to the characteristics of the API are really important in terms of being successful for development. So really what we're after to solve product development is to really create a strong product development team. And so it's the API, understanding the physical and chemical properties of the API in context of the dosage form. And then from the drug product side, understanding how the impact of the API properties and consistencies, understanding the process and how that impacts the process, and also the knowledge of the API in problem solving and troubleshooting. And again, this all revolves around having strong team infrastructure and team processes in place, so you're not, inventing or reinventing the wheel in order to get this accomplished. So really, to draw the whole box around us, this integrated analytical development knowledge basically takes the information from the API, works with the drug product, the team shares that knowledge and information, and you're basically able to advance products quicker and also solve formulation and process scale-up issues. So now I'd like to turn to a couple of examples where our integrated approach has really helped move products forward. I wanted to mention a little bit about project teams and why they're so important. The dosage forms that we're working on are becoming a lot more complex. Our dosage forms are multi-active products, multi-layered products, modified release, controlled release, targeted specific GI release, et cetera, and we're looking to maximize therapeutic benefit and minimize risk. So dosage forms are complex. Project teams are also important to understand quality by design attributes, which are required for regulatory submissions. Process understanding, design and control, how the API properties and flow interact with the formulation, and the knowledge of the physical and analytical chemistry of the product. And then finally, speed. Having an integrated product team that can anticipate challenges and problems can bring these novel therapies and medicines to patients faster. It also, for a startup company that's going through a burn rate, if you can get through a program faster, you can conserve more of your capital. And then finally, clinical trial scheduling. Sometimes clinical trials are scheduled seasonally, and making sure you have the product in the right place at the right time is critical. So basically, the analytical folks that focus just on end product testing really are missing some of the point. The integrated analytical understanding creates that scientific knowledge, and it's basically doing the right experiments, understanding the science, and that results in developing medicines faster. And also project teams, there's a great cartoon I really like about Gary Larson, where all of the horses are, and I guess it would be appropriate for Denver, all the horses are plastered up against the sheriff's office, and the caption is, "Jeb, you just can't throw a posse together, you got to organize it." And inside of my comment about project teams, you just can't throw a group of people together and say, "Hey, let's work." You have to have a supportive environment, which means the management expects it. You monitor their performance, you escalate processes. Senior scientific review is already functioning, so as the product develops, you have scientists and senior scientists with experience reviewing their strategy. Your project management systems have technology behind it, so you can understand capacity planning, resource management, status, and KPI performance about those projects. The project, there's shared goals, you understand the stage of development, what you're trying to accomplish, how that works with the customer. The project manager is responsible for staying objective, managing egos, and coordinating customers. Focusing on what is right to advance the product, not just who is right, and value all perspectives for problem solving. And in many cases, naive questions, typically outside of your discipline, can be the ones that are the most important. And the key is really understanding what the customer needs, not just what they ask for. Many customers say they want a certain thing, but you sometimes have to drill a little deeper and find out, okay, what is it that you really need, not just what you're asking for. So let me go through a couple of examples of integrated product development to show how knowledge in multiple parts of the project really fit together to accelerate development. I'll show an excipient compatibility studies for a delayed release formulation, and also talk about process development and manufacturing troubleshooting for two examples. So for excipient compatibility delayed release formulation, this was designed to be a delayed release tablet. In other words, a product that's released in the intestine, so bypassing the low pH of the stomach. This was a small molecule API with amine, primary amine and methoxy acid substituents. And there was some existing compatibility data, but the customer really wanted it expanded to include a potential enteric coating component. So here was an example where we had knowledge of the API, a desired dosage form, and then we designed studies around this. So we looked at potential excipients to be studied for a delayed release formulation. And so typically, excipients that were included as diluents, disintegrants, surfactants, what have you, and then looking at aqueous enteric coating, specifically cellulose acetate phthalate and polyvinyl acetate phthalate as enteric coating compounds, and then also looking at an OPA-dry as a subcoat polymer for tablet experience. So again, looking at a number of excipients that would commonly be used in a modified release, a delayed release formulation. And so basically this protocol set up pretty standard binary mixtures stored at accelerated temperatures, 40-75, 25-60 for up to 12 weeks, and testing at 4-week intervals. And importantly, included controls, individual components, and the API. And it was a fairly straightforward reverse phase HPLC determination with detection at 220 nanometers in the UV. So what did the data show? So what was interesting about this is that for all the mixtures, there was no detectable degradation product seen except for the mixtures of the API with polyvinyl acetate phthalate and cellulose acetate phthalate. And so these two excipients showed degradation products with similar but not matching retention times in the presence of the API, and the API alone showed no degradation. So it was interesting. So we had an API with an excipient was degrading, the API alone didn't, and then the degradation pattern of the excipients alone was different than when combined with the API. So really something here is going on. And so it turns out that there were amidation products from the excipients were actually reacting on the primary amine of the API. And so we looked at some of these chromatograms for 8-week samples, and it's interesting. Here's a sample of the API plus the polyvinyl acetate phthalate alone, the cellulose acetate alone, and then the API alone. So again, no degradation up there. And it turns out whenever you had one of the phthalates present, not surprisingly, you had phthalic acid degrade. And so in the presence of the API, these new peaks showed up. And basically these were amidation products resulting from excipients reacting with the primary amine on the API. So essentially you had degradation of the excipients which then reacted with the API. So again, it was putting this puzzle together. So essentially going back and figuring out what was going on, you looked at the chemistry of the excipients, understanding the chemistry of the API. So there were esters in these two polyvinyl and cellulose acetate, an acetate ester and a phthalate ester. And so these were both hydrolyzed to produce acetic acid and phthalic acid. The phthalic acid is separated and detected, and when it reacts with the API, that's also detected. The acetic acid doesn't really show up in the UV chromatogram, so you don't see that, but you do see a degradation peak associated with it. So here was a case where the knowledge of the API and the understanding of the desired formulation and the excipient components all pulled together enabled you to understand how this particular API would react in a delayed release formulation. So the conclusion from this particular study was that the API and the excipients do not react together. It was the degradation product of the excipient that reacted with the primary amine. These binary mixtures may provide the worst case as coatings may limit contact points, and you could consider using a barrier coat if you really had to use these particular excipients. So the point I wanted to make about this is the analytical development scientists were working closely with the formulation scientists to understand the degradation chemistry along with the formulation design. So again, that integrated analytical presented risks and options to the customer based on data. They may need a barrier code or different functional codings. And so this contrasts with just testing samples with a general assay method may not have developed this sort of knowledge of the interaction of the API and the formulation. So again, an opportunity to take API and formulation knowledge, combine it together and come up with a better product idea for a customer development. I'd also like to talk a little bit about an example with analytical development integrating with process development and basically using all of the tools you have available to understand the process. So this was a case where we had a single active. It was a small molecule API formulated into a film-coated tablet. There were multiple strengths of this product involved, 25 through 200 milligrams, with similar excipients but not exactly dose proportional. This was a wet granulation with intergranulate combination and then blending with other excipients. And feasibility experiments were done with all 4 strengths early in the process scale-up. And so here shows the data for these 4 different strengths, blend uniformity, content uniformity, a 30-minute dissolution test, assay, and moisture. And the part of this that was troubling is that we're getting some fairly low assay values for the 25 milligram and then increasing closer to what we would expect for the 100 and the 200 milligram strengths. And so why do we just get low assay results for the 25 milligram strength? Does this really make sense? And so, of course, I love being an analytical chemist because you blame the laboratory first and then you figure out what the real answer is. And so, of course, the question is it the assay method or is it actually the product? We're always at the end of this, so we're used to taking the pain of the formulation. But anyway, a very simple way to incorporate what we were working on is running an infinity time point with high-speed stirring. So again, the dissolution method was a little bit different than the assay. Really ramp up the speed at the end, make sure you're extracting everything out of the tablet, and then compare that with the assay. So we did that, and on this slide, the 60-minute time point represents something like 150 RPM, so it's really stirred very strongly. And we're basically seeing very little difference in the 30 to 60 minutes of the compounds, perhaps a little bit at the 200 milligram. But these assay values, while not exactly the same, are really lining up with the dissolution results. So here is a case where you've got 2 different methods basically correlating that this 25 milligram product is really off. It's really something's happening here. It's not the analytical method, something else is going on. And so this then allowed the team to focus on the manufacturing issues. And the team, the analytical chemists working with the process chemists, looked at the manufacturing train. They identified API losses in the granulation process and retention on filters. So again, there were parts of the formulation that were trapping this active ingredient, causing that tablet to be low. And so the team working together changed the blending processes and eliminated the losses and got the strength back up to what their target was. And the answer to this one was that there was no one single team member that had all the data and information to solve this problem. So again, it's integrating that analytical with our formulation colleagues to really solve the problem, and working together this problem was solved. So again, the example, the thing I'm trying to stress with this talk is really the criticality of having your analytical and formulation teams together, not sending off to another lab, not something remote or disengaged, but really having these colleagues work closely together to understand the problem and to create better formulations and solve problems faster. And basically, for effective development programs, the analytical development must coexist with the formulation, same lab, same site, same objectives. Team-based knowledge create scientific value for our customers, and then teams also need infrastructure, a common language, and management support to be successful. So again, thank you very much. This was my presentation for integrated analytical development at Patheon. Thank you.